Professor Suresh. He's from Division of Molecular Oncology, ICMR, NICPR, Noida, and worked in cancer biology and breast cancer. Did his PhD in molecular biology from Jamia Islamia, New Delhi, and has life membership of Indian Association of Cancer Research. Now I invite Professor Suresh to deliver. Yes, please. 25 minutes.
I will take 50 minutes.
No, take 25 minutes.
I will complete on the top.
Yeah.
Good morning to all. I am Dr. Suresh Hedau, NTC from the ICMR National Institute of Cancer Prevention and Research. It is located in the Noida. First of all, I thanks to organizing secretary Dr. Alok Chandra Bharati here giving me the opportunity to present my work. Here to explore the molecular signature and establish the drug resistance model in targeted therapy resistance in breast cancer by approaching the omics profile. As you know that the breast cancer, as earlier speaker it is already told that the breast cancer now it is rising in the metropolitan cities and today represents a significant challenge in the public health. So that the global incidence it shows that it is both excessive in the male and the female. So breast cancer is rising. Earlier that the lung cancer it is topmost, now the breast cancer is going up, followed by the lung, colorectal and the prostate cancer. The types of the breast cancer based on the tissue origin and the molecular signature. As you know that the breast cancer, that the invasive ductal and the lobular ductal carcinoma. Invasive ductal carcinoma has comprised many of the approximately 80% of the cases has shown in the invasive ductal carcinoma, and approximately 20 to 20% are showing in the invasive lobular and the other carcinoma. Here the luminal A and the basal A, it is the hormonal negative and the it is the HER2 positive and HER2 negative breast cancer cells. So here that the drug discovery, preclinical development, clinical development, approval and the drug challenge are the continuous process in the cancer management. Latest breakthroughs in the molecular biology and targeted personal therapies have been tailored to the specific pathophysiological condition in different types of the breast cancer. Tumor cells acquire drug resistance against the anti-cancer drug. Here we are lacking the knowledge of the molecular and the cellular events associated with the acquired therapy resistance. However, there is the clinical and the preclinical reports elaborates the different possible mechanisms of therapy resistance. Here it is the so many possible pathways are there. I am only concentrate on the few pathways. So first pathway is the stimulus induction. So the OCT4, SOX2, NanoG, CMYC and KLF4 are known stem cell transcriptional factors that involved in the regeneration and the maintenance of the stem cell characteristics. These transcriptional factors also modulate the tumor cells for metastasis and acquired drug resistance in stem cell population enriched in the tumor cells which generally detected by stem cell marker such as CD44, CD33, FCAM, gene promoter, aerobic glycolysis process, and suppress the oxidation of PCA cycle for uncontrolled and the proliferation and survival of the hypoxic condition. Overall, the transcription factor involved in the acquiring drug resistance phenotypes and the cancer progression. Epithelial to mesenchymal transition. In normal condition, epithelial cells are tightly interact and tight junction to adherence the junction and support the basal membrane. During the tumorigenesis, cell morphology changed and cells are loosened, the basement membrane are disturbed, and finally the cells acquire mesenchymal characteristics. So E-cadherin, cytokeranin, and some other are the mesenchymal markers to use to detect whether the cells are normal or it is a tumorous. This transformation induced by various signaling pathways, GR, RKT, TGF beta catenin, and Notch signal. EMT induced invasive spreading tendency, stemness character, and promote survival signaling and block apoptosis signal, and finally cells become tumorigenesis and acquire therapy resistance. There is a ROS mutation. Reactive oxygen species generate due to oxidative reaction mostly in mitochondria, and its concentrate in the cell decides whether the cell go to survive or undergo in cell death process via induction death signaling cascade. However, increased ROS level sometimes initiate the transformation of normal cells into cancerous cells and also promote the cell migration. Epigenetic changes in drug resistance tumor cells. Environmental factors play an important role in tumor initiation, progression, and therapy resistance that modulates the carcinogenic signaling cascade via epigenetic changes. This is the ABC transporter, is a very important protein involved in xenobiotic effects, including the anti-cancer drug. It elevates the expression observed in various cancer and tumor recurrence condition. So P glycoprotein, MDR1 and ABGC2 are commonly used as a marker for therapy resistance in various cancer. That is, the post-translational regulation of gene expression is very crucial phenomena in the modulation of cellular function. The characteristics of cancer cell as well as the drug response are highly dependent on modulation of signal transduction that is modulated by at the post-transcriptional level by mRNA. However, the drug response is vary for different drugs. That's why we are selecting the kinase inhibitor based targeted therapy. In my lab, we developed the drug. In our group, we are focusing the molecular signature and mechanism, signaling network, post-transcriptional and post-regulation, post-translation regulation, and many other possible events happening in the acquired drug resistance in breast cancer. In continuation of this study, we are looking the new drug target and new drug candidate to resolve the drug resistance challenges. We have developed the drug resistant cells by continuous drug treatment for several passages and evaluate the drug resistance phenotypes at cellular and the molecular level, and then go for the omics profiling. Integrated data analysis and outcome of the analysis data validated in the clinical samples. Finally, we will try to identify the molecular signature and new drug target in the resistant cells. In my lab, we have developed the drug resistance model in abemaciclib and palbociclib. These are the FDA approved drugs for the advanced treatment of the breast cancer, and they are target in the CDK4 and CDK6 is the kinase inhibitor, and we have calculated the IC50 value of both the cell lines MDA MB231 is the triple negative breast cancer cell lines, and the MCF7 it is the hormonal positive and HER2 negative breast cancer cell lines. Again, we have tested for the urolimus drug because I have recently got the project from the DHR. It is from the DHR funded. So we are working on the urolimus and the rapamycin. It is the mTOR target inhibitor. Ipirubicin, it is from the ICMR project. It is from the Hedau ICMR project. It is recently got it is after only for the 5 to 6 months back, and the work is ongoing on. And unique proteins and mRNA identifications as a molecular signature in abemaciclib, palbociclib resistance model in MCF7 breast cancer. And we found that some of the proteins are upregulated and some of them are downregulated in the abemaciclib resistance model in MCF7 breast cancer cell lines, as well as it is some of the proteins are upregulated and downregulated. It is in the palbociclib resistant cells. Some of them are in the upregulated. miRNA and gene identification urolimus resistance in MDA MB231 cells, and we found that the panel of mRNA identified as a potential prognostic marker in the urolimus resistance drug, and mRNA and gene identification in ipirubicin resistance model in the 231 cells, and we found that 11 unique genes identified in the urolimus RMDA 231 cells, and corresponding 31 mRNA identified that targeting the mentioned unique genes. Rather than these outcomes, we have more omics data of drug resistance in analysis process and have to wait to some concrete area. This is my institute, it is National Institute of Cancer Prevention, it is the ICMR Institute, and this is my team. Without the team, we cannot conduct the research. There is the 2 PhD students, 2 postdocs from the DHR, that is the DHR in scientist, and this is a small group.
Thank you, Dr.
Thank you, sir.
Thank you very much for your talk on the role of omics in looking into the drug resistance and also working on new strategies to treat the breast cancer. So we don't have time for questions, but again you can interact with Dr. Suresh after the session, and I now